So first, let's start off with what an organ transplant is. So as you all know, it's when someone has a disease organ and they need another organ from another person to replace it. But do you guys know what organs can actually be transplanted? Do you have an idea? Kidney. Kidneys. Okay. All right. Liver.
Heart and your lungs.
Heart and lungs.
That's two.
Intestines. Skin, right? Yeah.
Eyes.
Eyes, yeah. So you can transplant organs and tissues. So these are all the main vital organs that can be transplanted. The first transplant was done in 1954. It was a kidney transplant. Kidney transplants are relatively simple because there are only 3 connection points between the kidneys and the rest of the body. But then as time went on, they learned how to do more complex procedures and more complex transplants. But even though they learned how to do all these transplants, there's still a huge gap in the number of people who need transplants and actually receive them. So there are currently over 118,000 people waiting for a transplant right now. And by the end of this year, only about a third of those people will actually receive transplants. That's crazy. That's a huge number of people who need transplants and will not receive them and will die waiting for one. But why is there such a huge gap in this amount of people? There are lots of limitations that prevent you from being able to receive a transplant. And even if you think, okay, if everyone says I'm an organ donor, like I'll donate my organs if I die, not every death can lead to an organ that can be donated. You have to die in a hospital on a respirator so your organs don't stop losing blood or oxygen. There's also a huge matching process between the recipient and the donor to prevent rejecting the organ. So when someone receives an organ transplant, there's a huge risk that your immune system will start attacking that organ because those cells are not your own. And that puts the recipient in huge danger. So you have to match by blood type, height, weight, and other medical factors so you don't run that risk of rejection. There's also the allocation process that determines who can actually get the transplant or who will get the transplant. So if there are 2 people that are the same height, same weight, and both deserve this organ, but one person is a little bit sicker than the other, they'll get it over the other person. And that prevents a lot of people from getting the transplants they need. So what are researchers already doing to kind of supplement or help this problem? They're starting to print 3D tissues they're printing 3D tissues to supplement the tissues that we already have so they work better. And they're also making robots that work with your body and help the malfunctioning organ function more properly. And they're also doing research to make the matching process more efficient so there's not as much time between when the donor organ is ready and by the time that the recipient can get it. But those things are just helping the process. They're not fixing the problem. So what kind of things can we do to fix the problem? So how about producing artificial organs instead of just supplementing the organs that we already have? So I'm going to present to you 2 papers that I looked into on how they looked, how to create mechanical organs and biological organs that are new and artificial and completely not your own. So the first paper I looked at was called The Development of a Fully Automated Closed-Loop Artificial Pancreas Control System with a Dual Pump Delivery of Insulin and Glucagon, or artificial pancreas. Before I talk to you about the artificial one, I'll talk to you about the real one first. It has multiple functions, but the main function that we're going to be looking at is its function as an endocrine gland. So an endocrine gland secretes products directly into the bloodstream, secretes hormones that help the rest of your body function normally. So the pancreas specifically secretes insulin and glucagon, which maintain the blood glucose levels in your body so they're at a healthy level. And this has to do with diabetes. So diabetes is when your blood glucose levels are not maintained properly. So either your pancreas isn't producing the right amount of insulin or your body isn't responding to those amounts of insulin. So the artificial pancreas device was actually FDA approved just this past November, so it's pretty recent. But it's the first fully automatic pancreas. So the current treatments for diabetes require other people to be monitoring your blood glucose level and delivering insulin directly to you. But this one does it all itself. This diagram is the very complex version of what it looks like, how it functions. But this is a much simpler picture. So what it is this little device that has a bunch of input information. So it has your weight, your height, and your target blood glucose level. So it knows exactly how much blood glucose you should have at any given time during the day. It has two sensors that directly sense your blood glucose level. It takes data every few seconds and sends that data to the device so it knows how your body's functioning at that time. And it's controlled by this method that directly mimics how the beta cells would work in your body to test these levels. So it's actually functioning as if it were the pancreas in your body, it's just external. So this device is meant to infuse insulin and glucagon into your body when it's necessary. So here we have a patient study. This patient had diabetes, and up here you can see these are her blood glucose levels over the span of 10 hours. So you can see the rise and fall in blood glucose. So insulin, this is the insulin infusion rates over the day. Insulin is produced to lower blood sugar levels in your body. So, as you can see up here, at around minute 100, there was a huge spike in the patient's blood glucose levels. So then, the device immediately responded to that and produced a lot of insulin. There's a spike in insulin here to lower those levels, and you can see that it did that, it lowered the levels. And then later on in the day, around minute 300, there was a dip in the blood glucose levels. So the device produced glucagon, which is supposed to raise the blood sugar levels, and it did that. So it properly responds to the information that it gets from your body. Does that make sense? Okay, so let's just review. Can someone tell me why this is a better solution to what diabetes treatments are already?
You don't have to monitor it all the time?
Right, it's done all itself. So, and cons, what can be complications with this? What kind of things could possibly go wrong with this device? Yeah.
It's like big and external, so like you might like knock into it and break it.
Yeah, so it sits on your hip, so it could break. It's mechanical, so it could malfunction. Something could happen to the sensors. It might not function properly. So it's a great device, it's one of a kind, it's never been done before, but there are lots of things that could possibly go wrong. So with these mechanical external devices, there are lots of risks that are involved in them. So what can we do, what research can be done to go around this problem even more? So I looked into another paper that discussed biological ways of producing artificial organs. And this one's called Interspecies Chimerism with mammalian pluripotent stem cells, or combining stem cells from one mammal with the embryo of another to create a mixed species organism. That's kind of confusing, but we'll get there. So let me just talk to you about stem cells for a little bit. Pluripotent stem cells, or PSCs, are cells that have not differentiated yet. So when you're first conceived, you start as like a group of cells that are just cells. They're not hair cells, they're not brain cells, they're not bone cells, they're just all the same type of cell. And so over time, those cells start to develop in multiple different steps, and then they become ready to become those neurons, blood cells, brain cells, whatever. So this research has to do with these PSCs and the different steps that they develop into, and which steps are necessary to create the development of two organisms together. So that being said, what's a chimera? A chimera is one single organism that's made up of cells from two different species. So you might think of like cat-dog, half cat, half dog. Not quite like that. It's more of like a tie-dye animal, so it's one animal with cells interspersed of a different animal. So if you can see this picture up here, it's like a mouse with some rat cells in it. Does that make sense? Okay, so what they did this paper in kind of 3 steps. They tried to create 3 different chimeras, one mixing rat PSCs with a mouse embryo, one mixing rat PSCs with a pig embryo, and one mixing human PSCs with a pig embryo. All right, so the first step. They did mixing rat PSCs with a mouse. And this has already been done before, so they knew how to do it, they knew it could be done, but they just wanted to go over the process, see how it worked, and see what kinds of things that they could figure out from it to further their research. So look at these, throughout these presentation, I have like a bunch of these pictures. Pay attention to them because it'll tell you everything you need to know about the whole process. So they start with a mouse zygote. So like I said, the zygote is the first cluster of cells that are just cells. They haven't differentiated yet. So they have the zygote in a dish, and they inject some rat PSCs into it. So they just take the undifferentiated mouse cells, take some undifferentiated rat cells, and put them into it, and then grow the mouse. And it worked. As you can see in these 2 pictures, the red represents the rat cells, and the blue is the mouse cells. So there are red cells interspersed with the blue. So that shows that the rat is contributing to the actual mouse. But they did see, after 2 years of looking at this one chimera, they looked at the different levels of cells that were in each organ, and they only saw that the rat cells contributed up to 10% of each organ. So that's not a huge amount of cells that are contributing to the organ, but they're still there. Something cool that they did see, though, was that the mouse had a gallbladder, which is normal, but rats usually don't have gallbladders. So this means that the rat cells and the mouse cells kind of work together, turn things on in one and off in the other, to work together to create this organ. And the rat cells actually did contribute to the gallbladder, which it normally wouldn't have. So these 2 cells were working together to create one full organism. Any questions?
So when the stem cells differentiate, do they differentiate as rat cells, or are they put into the mouse's body or the rat's body? I forgot which, and then differentiate as the host species stem type of cell?
They're as their own. So like as you can see here, like it wouldn't be red and blue if it was different.
Okay, makes sense.
So it's like there's rat cells in the mouse. Yeah.
Julia, what is it about this process that's preventing the host's immune system from attacking these cells? Is it just that those cells were present when the immune system wasn't developed yet, or what's responsible for that?
I think it's what you're saying. They're kind of developing together. So there was never a time when they weren't together, so they didn't know that it was foreign.
And the big thing about it is they're trying to match the timing of development so that it doesn't affect them.
Yeah, so going along with that point, they did the next part of the study. So they tried to mix rat and mouse PSCs with a pig host. So they did the same process. They took the zygote of the pig and injected rat and mouse stem cells into it. But after 28 days, they were in the pig. They were developing normal, like as a normal pigs would. And then they took them out. They killed the pig, took out the embryos to study them, but they showed no contribution to pig embryos. So there were no mouse cells or rat cells that were in the pig embryos. And so this has to do with what Ms. Legerfo said is the timing of the development. So normal rodents, it takes them like 28 days to develop into a fetus to be born. So they're only in the womb for 20 days. Pigs, on the other hand, are in the womb for like 120 days. So it would take them a lot longer to develop than rodents. So that timing of development was off. So the cells weren't able to work together to create a chimera. Any questions? Next, even though that one didn't work, they decided to try human cells anyway. So they took the human stem cells and cultured them into 4 different steps of development. So here there are 4 different steps. The scientists learned how to use specific growth factors to stop the development of the stem cells at different stages. So they could see which stage would work best when working with another organism. So they took an egg of a cow and a pig, and they fertilized it and injected the stem cells into it, just like they had before. And then they cultured them for only 2 days. So they injected them into the blastocyst, which is like a structure in early development. So it's pretty much the same thing as a zygote, but it has this thing called inner cell mass. And the inner cell mass is what later becomes the embryo. So they were only looking at 2 days worth of development, and just to see if those cells would even work together and survive with the pig and the cow cells. So like I said before, they had the 4 different stages of human PSCs, and they looked to see how many of those cells remained in the inner cell mass of the blastocyst of the pig and the cow. And all of them worked, but the NHSM line worked the best. So that one had the most cells in the inner cell mass after 2 days. Make sense? Good. Alright, so then they tried to take another step. They wanted to look at the actual embryo, see the blastocyst develop into an embryo, and see how many human cells would be present in that embryo. So they decided to use the pig instead of the cow. Even though both worked, the pig anatomy is a lot more similar to human anatomy, so they figured that would be a better match process down the line. So they decided to look into the pig. So they did the same test. They took the pig zygote, they injected the 4 lines of cells into the zygote, planted them into the pig uterus so they could grow and develop normally. And then after 21 days, they terminated the pregnancy to look at the pig embryos. And they did see that human cells actually did contribute to the embryos of the pigs. So these are kind of confusing, but I'll break them down. So if you look at that graph over there, the light blue and the light orange are the embryos that contain human cells. So that just those 2 chunks are the ones that contained human cells, and the others, the dark orange and the dark blue, didn't have any human cells present in the embryo. So out of the 186 embryos that they harvested, only 67 of them actually had human cells contribute to the embryo. So then they looked kind of at the ratio of development in the embryos with human cells and without human cells. So both the blues represent those with normal development, and the orange is without normal development, with slowed down development. So as you can see in that graph over there, the embryos that were without human cells were more likely to be normal sized than those with human cells. So this kind of said, oh, okay, maybe there's something going on here. So then they looked at all of the embryos that had human cells in them, and they looked at the ratio between those that were normal size and those that had stunted growth, and they saw that the majority of them did have stunted growth. So this is kind of just showing that there is some difference between humans and pigs in their development, and they weren't fully able to make a functioning, fully developed chimera, but they made some. And again, this makes sense if you think about the gestation periods for humans and pigs. So like I said before with the rat and the pig, there was like a huge difference, and they weren't able to work together. So between humans and pigs, I said pigs were 120 days of development. Humans are usually at 280 days of development. So that could contribute to the reason why there were so many that weren't growing at a normal rate. But this is pretty awesome. They're taking human cells and putting them into another organism and creating embryos that had human cells contribute to them. So their main goal for this research is to create organs that they can use for testing that will better mimic human bodies. So a lot of the testing that is done in science now is done on rats and monkeys and pigs, and they don't have the same anatomy as humans do. They won't react to those drugs and those models just like humans do. So having organs like that had human cells in them would give them better results and more accurate results to what a human would react to those drugs. And also for diseases, to recreate diseases to study in organs with human cells is a lot more effective than studying them in rats that have different anatomies than we do. And of course, bringing back to the earlier part of the presentation, a main goal is to generate human organs to transplant into other humans. So instead of waiting for someone else to die for you to be able to use their organs, why can't we just grow those organs and use them to transplant to people to lower that gap between the people who can get transplants that need them? But there are some things to think about. What would it mean for us to be using other organisms just to put our cells into them so they can grow for us? Would that be okay? Is that ethical for us to use pigs for our organs? I don't know. Do you guys think so? Yes?
Yes.
You think it's ethical?
Yeah, I think it's okay.
All right.
We eat them. So
That's it.
Fred, you're
Thank
like-
you. This was.
I just want to make one clarification comment. Someone said that you can transplant eyes. You can't transplant the whole eye. You can transplant the cornea.
Anyone else? Yeah.
Is it like a significant risk for rejection with using another animal, like blood type and stuff like that, because it's not a human?
Yeah, so this is this is the first study that's ever been done into chimeras, so they have a lot more research that needs to be done, and there are a lot of questions that this paper raised that need to be looked into. So they're not quite at that stage yet where they're gonna put these organs into humans, because they didn't even look at the embryos past being embryos. They were never actually born yet, so they don't even know how they can function in a normal pig rather than a human yet. But I think that's kind of like where they're going in the future, where they hope to go in the future. Mr. Wise?
I have two questions.
Yeah.
One is for the pump that you showed us that releases the insulin.
Yeah.
What is the energy source? Is that a rechargeable battery?
I don't know.
It didn't say. And the second one is, how is that different from the current insulin pumps that are already on the market? Because I know I've treated athletes who have an external insulin pump where they don't have to be concerned so much about measuring their sugar levels all day long.
Okay.
So that insulin pump releases an amount of insulin. It didn't have glucagon.
Yeah, I
It's
think
only
that's what it,
That's the difference.
yeah,
So it's just one
I guess that's
medication
just
on
both.
both.
right.
Yeah.
Are you aware if there was any discussion or negative feedback in response to this paper with respect to the ethical issues mentioned?
I don't know. I didn't really look into it. I could imagine that there would be, just because of the implications of it. I don't think it was as publicized as it would have been if it was done like further research into it, because it was just done in embryos, like I said, like it wasn't full organisms yet, but I can imagine that there will be in the future.